Research is the cornerstone of innovation in clinical practice, driving improvements in patient outcomes and shaping the future of medicine. Welcome to the Global Rhinology Network Topical Research Symposium. I'm Ashok Rokade from Winchester, UK, and it is my privilege to welcome you as a host. Joining me as co-hosts are Louise Melia from Bath, UK, and Professor Thibault Van Zele from Ghent, Belgium. This symposium showcases fascinating topics across 4 key modules: General Rhinology, COVID session, and Research in Chronic Rhinosinusitis. A special highlight is the PhD session featuring research conducted by enthusiastic young rhinologists who represent the future of our field. This symposium brings together leading global minds in rhinology who will highlight latest developments with a focus on translating research into impactful patient care. The Global Rhinology Network is dedicated to advancing knowledge innovation. Thank you for being part of this exciting journey. Thanks to Medtronic and Karl Storz for their generous support in making this symposium possible. Welcome to GRN Topical Research Symposium.
So let's go ahead for today with our first speaker, which is actually a very good colleague from me from University Catholique de Louvain in Brussels. And I would like to introduce to you Professor Valerie Hox, which is an academic rhinologist who works in the department of ENT at the UCL hospital in Brussels, doing rhinology and anterior skull-based surgery. But also she dedicates half of her time to translational rhinological research, mostly focused on unraveling the pathophysiology of chronic sinusitis. So please, Valerie, share us your latest insights and results of your research.
Thanks, Thibault, and everyone else for this introduction and for inviting me. Can you see my slides?
Yes.
Okay. It's really an honor to speak in this really nice symposium with really nice speakers. Unfortunately, I'm in the midst of a clinic, so I'll have to run away afterwards. I cannot stick around, but it's really a nice program. So I'm very happy to show you a little bit today about what experimental models are available to study disease mechanisms of chronic rhinosinusitis, and especially those that we are currently using in our lab. So I don't really think the audience needs an introduction about what is chronic rhinosinusitis. You all know that it's a disease characterized by an inflammation of the nose and the sinus mucosa, leading to the typical symptoms of nasal obstruction, secretion, headaches, and loss of smell. And in a certain percentage, this comes with nasal polyps that are visible in the nasal cavities. When looking at pathophysiology and research that has been performed in the past, we see that most of the research has been gone to eosinophilic chronic rhinosinusitis, which is in general with nasal polyps. And the research on this topic actually has exploded in the last couple of decades, and a lot of findings have been published, which is really good because this actually led to the discovery, but also even the implementation of novel therapies that are directed towards certain cytokines and mediators. However, on the side, when we look at the non-eosinophilic counterpart of chronic rhinosinusitis, which is generally a form that doesn't present with nasal polyps, we see that it's still some kind of black box and very little is known about disease mechanisms. So what can we do to gain more insight in this non-eosinophilic chronic rhinosinusitis, but also still in the eosinophilic group, of course? Well, we like to do in the lab what we call translational research. And translational research is combining different levels of research. So we all know that the most valuable data is coming from the clinical trials, where we can harvest human samples for observational and interventional in vivo data. But in addition to this, we can make use of animal models and also cell culture systems to study disease mechanisms by overcoming certain limitations that are linked to clinical research. So most of you are very familiar with clinical trials, I think, so I thought I might focus a little bit more on animal models and cell culture systems that we have available to go a little bit deeper in these disease mechanisms. So when talking about animal models, I'm going to focus today on mouse models, specifically of chronic rhinosinusitis, which have some clear advantages compared to human clinical studies. For example, we can try different interventions without overcoming those difficult ethical committees. We are working with a homogeneous population from the same genetic background, the same environment, the same food, instead of working with humans who are very heterogeneous, of course, and go home after you see them at the consultation and they do a lot of things you don't know about. With mice, we have the possibility to work with thousands of transgenic strains that are available, and this is probably the most important advantage. So we can very easily find out the mediators that are involved in a certain disease. And there's also a very wide availability of reagents and antibodies directed or purified in mice. So we can do a lot of experiments in the lab. Also, it's important to note that there do exist a lot of similarities between human and murine immune responses, and 85% of the encoded proteins for the immune system are similar between humans and mice. However, on the other hand, we have, of course, some negative points. The obvious ethical burden that is related to animal research, the fact that the sinuses of mice are smaller and different compared to humans, slightly different. It's also hard to obtain chronic disease unless you want to do a mouse model of several years. And by definition, every disease that is induced in a mouse is artificial and is bypassing natural pathways. And last but not least, unfortunately, mice have been shown to be poor predictors of therapeutic response, because only 2% of the treatments that work in mice actually make it to the clinical trials. However, there has been some research that has been done on mouse models, which is mostly focusing again on the eosinophilic chronic rhinosinusitis with nasal polyp group. So you all know also by now that it's an eosinophilic disease in the most severe forms that are most common in the western part of the world, with the presence of activated T helper 2 and innate lymphoid cells, and the presence of the typical cytokines being IL-5, IL-4, IL-13. And when looking at causal factors, there is still a lot of question marks remaining, but there is one major factor that comes back in almost all the research, and this is shown very nicely in this already pretty old paper from Thibault, who shows that Staphylococcus aureus colonization is linked to the presence of nasal polyps versus non-nasal polyps. And it's also linked to the severity of disease. And not only the presence of this bacteria, but more specifically also the response of the body to its superantigens, for example, Staphylococcus enterotoxin B. So when developing a mouse model, researchers have chosen to expose mice to an allergen to induce an artificial eosinophilic disease and to combine to this exposure to Staphylococcus enterotoxin B. So you end up with a mouse model that looks like this. There are certain variabilities to this mouse model, but they're all based on the same principle. You start with a sensitization phase to an allergen. This can be an artificial one or a real one, such as house dust mite or fungi. Then mice are challenged to the allergen, followed by a challenge phase to the allergen and the enterotoxin. And then after 12 weeks of exposure, the idea is to develop an eosinophilic chronic rhinosinusitis with nasal polyp disease. So when you look at the outcome measurements of this mouse model, you can see when we look at the maxillary sinuses that you see a lot of similarities with human disease. So there is the epithelial disruption and thickness that you can see here, which is also seen in humans. We see a goblet cell hyperplasia with an increased mucus production in eosinophilic CRS mice. There is epithelial fibrosis and basal membrane thickening. And last but not least, of course, we see the typical eosinophilic infiltration in the subepithelial and intraepithelial space in these mice that are co-exposed to the allergen and SCB. When looking at the nasal lavage, you can see that this is confirmed by the eosinophils that can be found back in the nasal lavage. And that very interestingly, we also find an increase of IgE in the nasal lavage, although levels remain pretty low because it's a very diluted sample that we get. But we see the same in serum. Although there is not really a difference between allergen and allergen with SCB, there's also an increase of IgE production. And interestingly, when we look at the lower airways, we also find an increase of inflammatory cells in the lower airways, which are also mostly eosinophilic. We personally did not look at the cytokine production in the nasal lavage, but the paper that firstly described this mouse model actually also showed an increase of type 2 cytokines in nasal lavage. However, there is one small problem related to this mouse model, and this is actually the generation of nasal polyps. Here you can see a coronal slide of a mouse skull that has been treated with ovalbumin and SCB, leading to all the features that I just told you. But when you look at the maxillary sinuses, you clearly see a difference for those who are used to look at CT scans of humans with the human situation. So in humans, you see the big nasal polyp formation filling up the whole sinus space, protruding into the nasal cavity. However, in the mice, it looks a little bit more like this. So it's only microscopically nasal polyp-like lesions with a little bit of edema, some eosinophils there, and some bubbly form of the epithelium, but they are very scarce and they are clearly not as big as in the human situation. And these are our results, only a couple of polypoid lesions per mouse, but it corresponds to what other authors have found in literature. So when we summarize this mouse model, you can see that there are a lot of similarities between human and mice. There is the epithelial disruption and thickening with goblet cell hyperplasia, basal membrane thickening, eosinophilic infiltration and IgE production with type 2 cytokine production, and united airway disease. However, the big edematous nasal polyp formation is very hard to reproduce in mice. So we can conclude that we have an adequate model to study disease mechanisms of eosinophilic chronic rhinosinusitis, but not really of the nasal polyp formation. So then there is also the second type of chronic rhinosinusitis, which is the typically non-type 2 or neutrophilic chronic rhinosinusitis, characterized by different kind of cytokines, IL-1, IL-6, IL-8, TNF, interferon, and the activation of T helper 1, 22, or 17 cells. But when we look also to the causes that have been published on non-type 2 CRS, it's also very hard to form a homogeneous population to this. And the causes that have been postulated are very variable. For example, there is exposure to irritants that has been proposed, the presence of biofilms. There is the structural problem with osteal occlusion and the consequent hypoxic damage. And finally, the exposure to bacteria, viruses, and even allergens. So when developing a mouse model, researchers were thinking of combining two of these factors, which was the exposure to bacteria in combination with osteal occlusion. The initial model where this model was used was actually a rabbit model, which was mostly used for surgical research. And here, researchers used a combination of mechanical occlusion of one nostril with a nasal tampon in combination with a bacterial or fungal inoculation. And what we did was we tried to translate this rabbit model, to a mouse model with obviously more difficult sinuses to work with because they are smaller, but with all the previously mentioned advantages. So we performed nasal surgery on day 0, where we opened up the skin of the mouse. We drilled away the nasal bones. We removed the nasal septum. We introduced pre-cut nasal packing, which is actually a tiny merocell, which is Pope wick, which is used for the ears. And then this tampon is inoculated with either saline or a bacterial solution. We chose to try Staphylococcus aureus, Streptococcus pneumonia, and Pseudomonas aeruginosa for their obvious link to chronic rhinosinusitis. And then mice were stitched up and they were sacrificed at either 4, 8, or 12 weeks after surgery to check whether we could induce a neutrophilic CRS. And what we saw was rather interesting. We saw that in the group that was affected with Staphylococcus aureus, the bacteria actually stayed present until 12 weeks after surgery in a standard or in a stable concentration, which was not true for Streptococcus pneumonia, which seemed to be cleared much more easily. And Pseudomonas aeruginosa was somewhere in between. In some mice, it stayed present, and in others, it was cleared. And this was confirmed on histological slides, where you could see that the bacteria are still present on and in between the epithelium. When we look at the nasal lavage, we could indeed see that at all different time points, there was an induction of inflammation, and all inflammatory cells basically were neutrophils. And this was mostly true for Staphylococcus aureus, and also to a lower extent for Pseudomonas aeruginosa. This was again confirmed on the histological slides, but mostly for Staphylococcus aureus, where we could find positive stainings for Ly6G. When we looked at the similar variables that we looked in the eosinophilic rhinosinusitis mouse model, we could also detect an epithelial thickness and subepithelial fibrosis in the groups that were treated with Staphylococcus aureus and Pseudomonas aeruginosa. We did some preliminary cytokine measurements where we could find most of the cytokines undetectable with a regular ELISA system, but we could see some trends of increased IL-8 TNF-alpha in mice treated with Staphylococcus aureus. So we're using a different technique now to confirm this. And also very interesting, when looking at the lower airways, we could also detect the presence of inflammatory cells in the bronchi, which were also neutrophilic, although there was at not one point a connection of the fluid, the bacterial fluid, with the lower airways. So when summarizing, we could see that we also could find some similarities between the mouse neutrophilic CRS and the human situation, with epithelial disruption and thickening, basal membrane thickening and fibrosis, infiltration of neutrophils, probably some cytokine type 1 production, and also the presence of united airway disease. So we ended up with an adequate model to start exploring disease mechanisms of certain forms of neutrophilic CRS, because I think we can all agree that this remains a very heterogeneous population and probably consists of different subtypes. So I know my time is running up, but I just wanted to quickly touch on the cell culture systems that are available, because they have some advantages towards murine models. They give better therapeutic response predictions. You can measure individual cell responses, and they're easier if you want to study chronic inflammation. However, there's, of course, the absence of a dynamic immune response where there is the interaction between the different cell systems. And you can see sometimes differences which are due to culture techniques and the media used. In the early days, people mostly used nasal epithelial cell lines, which were coming from a squamous cell carcinoma from the nasal septum, which were very easy to grow and reached a certain differentiation state. But of course, due to their neoplastic origin, they gave different growth patterns and inflammatory responses. So in general, we gave up on those. So then most of the people who are doing research transferred to the primary nasal epithelial cells, or NECs. And they are actually freshly isolated cells from biopsies, where epithelial cells are isolated through pronase solution, then negative selection of the epithelial cells. These cells are plated and then put in what we call transvals. For the first days, they are put all in liquid, in medium. After 7 to 10 days, we remove the apical medium to create what we call an air-liquid interface, and this actually gives rise to the differentiation of the cell line, leading to a nicely fully differentiated epithelium. As you can see here, with the presence of basal cells, some cilia, and the presence of goblet cells, which has as an advantage that you get fully differentiated cells, and you can even perform co-cultures while putting other cell types in the lower compartment. However, they're not so easy to grow. It's a very long protocol, And there's only a short time window where you can expose your cells to certain factors or mediators or disease mechanisms. And you can only use one well per test. And also they're based on a stiff porous membrane, which is sometimes difficult to cut. So that's why people are looking for different organ systems, such as, for example, now the 3D cultures or organoids, which are harvested in the same way, so by biopsy or even brushing now. Epithelial cells are isolated, expanded in a flask if necessary, and then they're seeded in a droplet of matrigel. And then when the medium is changed, these cells also differentiate. And when you look through the microscope, it looks like this. You have these different floating spheres of nasal epithelial cells, which when you perform H&E staining, they look like a multi-layered epithelium with a lumen in the middle. And we didn't do the stainings, but this is a very nice picture of the group of Sarah Vreugde, who is also presenting later on. And they could show that these organoids actually express markers of fully differentiated epithelium, such as MUC-A2, tubulin of ciliated cells, and also the tight junction stainings. So the advantage here is that you need less material. Even with brushings, it's easy, and they can be biobanked because they're much more resistant to freezing and thawing. On the other hand, there is still little experience with these organoids, and the future will have to tell us whether they will be more beneficial than the HNICs. So in summary, we can say that clinical trials are still the ideal way to study disease mechanisms and especially therapeutic effects. But there are, of course, some limitations, mostly due to ethical issues and population heterogeneity. And in that case, we can use, for example, animal models to study disease mechanisms. However, there are some differences between human and mice, of course, and then we can move to cell culture system, which can be available in 2D and 3D. And at this point, algae cultures were the workhorse, but now maybe organoids form a promising new model. Okay, this is what I wanted to tell you. I'm sorry if I went over time. Wanted to quickly thank my colleagues at the Lab of Pulmonology and ENT, and especially my students who did the work with the mice, and Cyprien who did the work with the organoids. So I'm happy to take any questions now, if possible.
Thank you, Valerie, for this excellent presentation. I think having a animal model for chronic rhinosinusitis, it's a little bit the holy grail. We talked in our group a lot on whether some animals like mice and rats and rabbits even can develop polyps, because if they would die, I think. What's your idea about that?
That's true, because they're supposed to be obligatory nose breathers. Although we did see some cases where we kind of induced a pretty severe nasal blockage, and these mice could still survive. So maybe if we could develop nasal polyps, they would survive if it's going slowly. But probably you're right, and they're not able to develop them. This can be due to several things, because the maxillary sinuses are a bit differently developed. The ostia are bigger, so it's more difficult to get osteal occlusion. The immune system is also slightly different, and indeed they're breathing in a different way. And then there's, of course, the fact that we use a mouse model of 3 months, and in general, nasal polyps probably in most cases take more than 3 months to develop in humans as well. Mm-hmm. So this indeed remains a very difficult issue in translational research.
Thank you. I have a question. Can I ask a question?
Yes.
Sarah? It's Sarah here. Hi. Very nice presentation, Valerie, and nice to see you for the first time. Start my video here. Yeah. Oh, sorry. Yeah, I just had a question about the bacterial strains that you put in your mice, because it's known that, for example, for Staph aureus, at least, mice, they lose, when you give Staph aureus to mice, they will lose certain virulence factors. So there is a certain prophage in there that is full of virulence factors that are lost. Did you do any kind of sequencing at the end of the experiment to look to see whether the Staph aureus that you got out of your mice is the same as the one that you put in?
We didn't, and this is actually really interesting, and this is also what I wanted to talk to you maybe about at some point, because we are, I know you have a lot of experience with the microbiome, so for us this is rather new, and we didn't compare the Staphylococcus aureus before and after, but it's probably a very interesting suggestion to do this to see whether there are some changes in these two strains. Thanks
I think that can be a real problem with looking at the effect of Staph aureus, because that's known in animals in general. They lose this kind of group of virulence factors that are essential, well, that are very often present in human Staph aureus, and so that's. But we can talk about this.
Okay. Later. Thanks.
Yeah.